and we'll chat soon. Thanks again for watching. Now, next lesson, I'm just kidding. Could you imagine if I did the lesson backwards? That would be so weird. Hey Bio20s, I am back again for another lesson. Today, we're going to talk about factors that impact enzyme activity. Okay, so we've been talking a lot about enzymes. So what an enzyme is it's essentially a protein that helps break down other things. You know it's an enzyme because of the ending ASE. ASE. Now, not all enzymes have this, but it's a really good indicator for most. For example, carbohydrates, amylase, maltase, lipase, protease. These are all things that have A's at the end. They help break things down. Now, why do we even need these things? Well, generally speaking, without enzymes, these reactions that we've talked about for the last few videos, they would take so long to occur that we would never get the nutrients we need. So enzymes are a must in order to allow us to help break things down. Because generally, the way you speed up reactions, you can use a catalyst, right? You can use an enzyme, you can increase the temperature. We probably don't want to increase the temperature too much in our body or else it would cause some major damage. So instead, we're going to use something called the catalyst, or in this case, an enzyme. So it's going to increase the rate of chemical reaction. The substrate is any molecule that is being changed during the process, and the active site is where the substrate and the enzyme bind together. So here's an example. We've talked about this one before. This is the enzyme maltase. So maltase there, that's the enzyme in blue. The active site fits perfectly with the substrate maltose. Maltase would break down the compound maltose. Maltase wouldn't break down anything else. So each enzyme is built or made specifically for the substrate it is going to break down. So for example, maltase wouldn't break down a protein. It would just break down maltose. And they fit together nicely, just like that. Now this was first discovered all the way back in 1973, where they introduced made all the way back in 1890. That was the first one. It was the lock and key model, where they figured out that, hey, each enzyme fits with the substrate, much like every key has a lock that it will open. Then in 1973, they made what's called the induced fit model, which is a slight improvement. Yes, every enzyme has a very specific substrate they can fit with, but you can actually alter the shape of the enzyme slightly to make it fit better. And how you do that is you can use things like coenzyme. You might remember coenzyme A from our previous videos of cell respiration, or cofactors. These are things that bind to the enzyme to make them function better. So here's a good example for you. In your red blood cells, you have an enzyme called hemoglobin. Hemoglobin is a protein found in your red blood cells to help the red blood cell bind to oxygen. One way to make hemoglobin more effective is to use iron. Iron is a cofactor that will bind to hemoglobin and make it much better at trapping oxygen and binding oxygen to your red blood cells. Okay, so how do we go ahead and change the ability of an enzyme? How do we make it faster? How do we make it slower? One way we can do that is by changing the pH. So the pH of a substance can be low, it can be acidic, it can be high, it can be basic, or it can be somewhere in the middle, it can be neutral. Enzymes function best in certain pHs. So let's take a look. Two enzymes found in our body, we have trypsin, we have pepsin. These are both protein enzymes. These are both enzymes used to break down a protein, and yet, if you look at pepsin, its optimal function is a pH of about 2. Will it work in 3 and 1? Yes. Will it work at 5? No. It's too, not even basic yet, but it's too far down that pH scale for pepsin to function. Look at trypsin. Trypsin, which is found in your small intestine, functions best at a pH of 8. Will it work at 9 and 7? Yes. Will it work if it's too acidic? No. So certain enzymes function best in certain pHs. If we change the pH too much, we change the structure of the protein, the structure of the enzyme too much, and it won't function optimally. So we want to make sure that it's an optimal pH for that enzyme. The second way we can speed up an enzyme is to cause more collisions. And this one's probably the most basic of any of them. If I add in more substrate, there's more things to break down. If there's more things to break down, there's more collisions with the enzyme, there's an increased rate in reaction. So if I wanted to increase the reaction rate of maltase, I would just add in more maltose. That being said though, you do reach a certain point where it will start to level off. So if no enzyme was used, here's my reaction rate, pretty small. If I was using a reaction, it goes much faster, but then it reaches a point where I can keep throwing in as much maltose as I want. Maltase is going to kind of level off a little bit, right? Because there's only so much enzyme available. Now, if I had more enzyme, that would change the story. But if we're just talking about substrate, eventually you can add as much substrate as you want. There's only a limited amount of enzymes available. But that being said, the more substrate we add, the increase in reaction rate we'll see initially. The next thing we can do to change an enzyme's activity is to change its temperature. Generally speaking, higher temperatures lead to higher reactions, but that's not always the case with enzymes. Enzymes will function best in a range of temperatures. So, if we change the temperature too much, we can actually change the enzyme too much, denature it, and cause some pretty severe impacts. This is why a prolonged fever can be very dangerous for the body. A fever is where the body's trying to essentially turn up the temperature, get rid of whatever's making you sick, but in doing so, if the fever lasts for too long, you can start to hurt your own proteins, your own enzymes, which can be a problem. So let's take two different enzymes. We've got an enzyme in a human and an enzyme in a thermophilic heat-tolerant bacteria. The human enzyme, you'll notice, functions best at an optimal temperature of about 37 degrees Celsius, which makes sense because our bodies are 37 degrees. Now, will it still function within this range? Yes. But we reach a point of like 50 degrees where it will no longer function. Our human enzymes inside of our body will only function within a certain range. If you look at a bacteria that's thermophilic, so once again, heat resistant, its optimal temperature is about 78, 79, kind of around 80 degrees Celsius. Way different than a human enzyme. They're both enzymes, they're both proteins, but they function in different temperatures. And then the last way to affect the rate of an enzyme is to use what's called a competitive inhibitor. If we use a competitive inhibitor, it can actually come in, block the substrate site, and block the enzyme from working anymore. So here's my enzyme, binds to my substrate, once again fits very nicely like two puzzle pieces. If I bring in a competitive inhibitor, which is another substance, it will block the enzyme from working and essentially slow it down. In a sense, turn off the enzyme's reaction. Now, the last thing I want to talk about is what's called feedback inhibition, which is how we shut off our enzymes. We don't want our enzymes running forever. We eventually want to turn them off, much like we want to turn off a light switch when we're done with them. So what we end up doing is when we've made enough product, it will actually feed back to the start of the metabolic pathway and turn that pathway off. And here's how it works. You take your enzyme binds to your substrate, perfect, nice, perfect fit. But you see this little site at the bottom here? This is the little part here where the final product, let's say it's glucose, can come in and bind to this enzyme. And you'll notice over there, when glucose binds to the enzyme, it changes the shape of the active site. So now the enzyme doesn't work anymore. It's essentially how we turn off the enzymes. If we need glucose, boom, we don't have much of it. So enzymes, they're working, they're breaking down your carbohydrates into glucose. Once you have enough glucose, you'll produce the glucose, bind it back to the enzyme, change its shape, and prevent the enzyme from functioning as quickly. So that's what we call feedback inhibition, where the final product will feed back to the start of the pathway and essentially turn off the enzyme and turn off the pathway. Another example of this is called allosteric activity, is where we take our initial reactant, we can actually use this to turn on an enzyme. We actually change the shape to make it function better. In either case, we're just changing the shape of the active site to make it either bind easier or make it harder to bind. All right, so that's it for today. Next video, we're going to talk about the respiratory systems. We're going to move on to our next video, or next system, which is going to be the respiratory system. And we're going to talk about the parts of the respiratory system and how those different parts work to get oxygen into your cells and get CO2 out of your cells. So thanks again for watching, and we'll chat soon.